Hello and welcome everyone to this podcast for genetics. This will be the last podcast for the series of podcasts on eukaryotic gene expression. And in this podcast, we're going to talk about my favorite field within genetics, and it's called epigenetics. It was first described in 1942 by Conrad Waddington. Let's think about this root word of epi. Epi comes from a Latin word for above and over. So epigenetics is a field that goes over or above or beyond in some way our classic thinking of what genetics is. And as you'll see, it does change a lot of the way we view genetics. In general, we can think of epigenetics as one of several mechanisms that result in a change of gene expression. However, this change does not alter the DNA sequence. So epigenetic changes are not mutations to the DNA sequence. However, epigenetic changes are passed on from one cell to the next, and in many cases from one generation to the next. However, they can be reversible. So they will get passed on from generation to generation, but this change in gene expression can at some point be reversed. But one of the most important features of epigenetics is that they are not gene mutations. So over the next several whiteboards here, we're going to talk about, in brief detail, the molecular cause of epigenetics. And then we want to talk about some examples of how behavior and environment can affect epigenetics. We'll talk about several examples of this as well, either in this podcast or in class. We'll touch a little bit on twins, something called X inactivation, and we'll come back to X inactivation at the end of the semester as well. And then the last thing, which is a really cool thing, one of the most interesting things of epigenetics, and that is something called genomic imprinting. So let's go ahead and get started with the molecular causes of epigenetics. Okay, let's talk about these molecular changes associated with epigenetics. The first mechanism I want to talk about is DNA methylation. Now remember, we don't mutate DNA with epigenetics, but the bases can be modified. So we're not changing it from, say, like a cytosine to a guanine or a thymine to a cytosine or anything like that. We take the actual bases and we add a methyl group to them. And methyl groups aren't just added to anything. They're added very specifically to cytosines. So if we have from 5' to 3', say a CG, AT, CG, and then of course the corresponding one below here, GC, TAGC, methyl groups are added specifically to the cytosines and only the cytosines. Typically, these are added to cytosines in promoter regions. In these areas where methyl groups are added, this will cause downregulation of the gene. So as a general rule, we can state that methylation causes decreased gene expression. I should also mention that the enzyme that adds these methyl groups is called DNA methyltransferases. And we often just call them DNMTs. DNMTs add methyl groups and shut down gene expression. Counter to this, demethylation. And demethylation is caused by enzymes called demethylases, which remove methyl groups and can increase transcription. But typically, once a region is methylated like this, it'll stay methylated for several cell divisions and in some cases can be passed on from one generation to the next generation of individuals. We see examples of DNA methylation linked to various things such as diseases like cancer, and DNA methylation explains how queen bees are selected to become queen bees. And I believe we'll talk about this example in class. The next mechanism I want to talk about that is linked to the molecular changes of epigenetics is histone modifications. So number 2 here, histone modifications. And some of these we've already talked about, but we'll mention them again here in passing. The first is histone acetylation. And remember, we said when a histone is acetylated, it removes it from the DNA, and so this leads to increased gene expression. When we talked about this before, I didn't talk about it in terms of epigenetics because we had not yet defined epigenetics. But when a histone is acetylated, it will often remain acetylated for several generations and will cause this increase in gene expression. Another modification of the histones is methylation. Now this varies what the effect is. So what I want to say here is some methylation causes increased expression. But if you methylate on other parts of the histone, sometimes this methylation causes decreased expression. Now remember, this is different than DNA methylation. DNA methylation results in decreased gene expression. Methylation of the histone, of this protein, can have different effects depending upon which amino acid is methylated in the histone. All right, that's all I want to say about histone modifications. And that's all I want to say right now about the molecular changes of epigenetics. So we've defined what epigenetics is. We've talked about the molecular aspects of epigenetics. But now I want to talk about what causes epigenetic modifications. And in this discussion, I want to talk about several things. That is, several examples. I want to talk about behavior, the environment, how that can cause epigenetic changes. I want to talk about twins. I want to talk about X inactivation. And then finally, I want to talk about something called genomic imprinting. So first, let's talk about an example of behavior. All right, so with behavior here, there's several examples, but I want to talk about one in particular that's pretty interesting. And it's a study where they took mice. So they took two groups of mice. Here's my mouse, as awesome as it is. And this other population of mice. And in one population of mice, the mothers did what most mice mothers do, and that is they groomed and licked their mouse pups, the baby mice. In this other population of mice, mothers did not groom or lick the mouse pups. And what they discovered was when they looked at these mouse pups, so the pups here had different patterns of DNA methylation compared to the pups who came from the mothers who did not groom or lick the pups. This ultimately affected the expression of genes that allowed these pups to deal with stress better. So pups in this group here made them less fearful and stressed, less stressed as adult mice, compared to the pups who came from the mothers who did not groom or lick them. So this behavior of mother mice caring for their young has a direct correlation to which genes get methylated and ultimately how the progeny, in this case the mouse pups, can deal with events in life as an adult. There are also comparative studies that have been done in humans as well, suggesting maternal care can have an effect on which genes will be expressed and how that affects the progeny as they get older. All right, so let's move on to environment. And in particular, I want to talk about this fungicide called vinclozolin. What they did in some studies here is they took a mouse and they fed it this vinclozolin, and I'm just going to write VIN. And this resulted in a male mouse, with reduced sperm production. Now this makes sense because vinclozolin will bind to androgen receptors, which prevents testosterone from binding to these receptors. And by blocking this receptor from testosterone, it prevents some of these male sexual development processes, such as sperm production. So this made sense. And this alone isn't really an epigenetic phenomenon. But what is interesting is when they took mice from this mouse, so progeny mice, we'll draw our famous mice here, and these mice now were raised without vinclozolin, and they too had decreased sperm production. So what caused decreased sperm production in this first generation was caused by the vinclozolin. because of the competition with the testosterone receptor. However, these mice here, they were raised without vinclozolin present, so there should be none of this blocking of the testosterone receptor from occurring. So why is this happening? In fact, the mice that resulted from this generation also had reduced sperm production. It went on like this for generations. And so when they studied these mice, they determined a few interesting phenomena. That is the progeny from each of these generations. One, no mutations in sperm production genes. So you might say, well, maybe the vinclozolin just damaged the DNA, mutated the DNA, and that's why this was happening. But they didn't see any mutations like that. But what they did see was increased methylation in genes linked to sperm production. So we didn't mutate, this drug didn't mutate the DNA, but it did change the expression of important genes for fertility when the original mouse had this chemical exposure. So an interesting question is why? We're not going to discuss that in this podcast, and we don't really know the answer, but it's a fun thing to debate about, is why would this be selected for throughout evolution? What is the advantage, or is there an advantage, for mice to have reduced sperm production generations later after an exposure to a chemical? Is there any kind of selective advantage? All right, now let's move on and talk just a little bit about twins. We're not going to say much about them, except talk about a study done with identical twins. And by their scientific name, which is monozygotic twins. Mono for one zygote. They arose from one fertilized zygote. We know that identical twins are genetically identical. Some exceptions, but in general, identical twins, monozygotic twins, are genetically identical. However, if you've ever met identical twins, indeed they do often look amazingly similar, but we do see some phenotypic changes. And what some studies have shown is that if you take monozygotic twins at birth, they have near identical DNA methylation patterns. In addition, their acetylation patterns also near identical. Now, if you take these same monozygotic twins and look at them years later, there are large scale differences in methylation of the DNA and acetylation. Start off with similar epigenetic patterns, but throughout the course of a lifetime, likely because of certain environmental changes or behavioral changes, there are epigenetic changes that result, which causes differences in phenotypes. Again, there are no mutations, it's just changes in the way genes are expressed. Okay, the next thing I want to talk about is X inactivation. X inactivation. So let's take a newly formed zygote. A newly formed zygote that is chromosomally XX. So it's a female zygote. The X chromosome contains thousands of genes on them. And so let's just look at two genes here. In fact, let's look at the colorblind gene. And let's say that in this gene, on this X chromosome, there's a mutation. And this gene, there are no mutations. We haven't talked about recessive or dominant traits yet. We mentioned them briefly, but we'll go in greater detail later on. But you may remember that as long as you're heterozygous for a recessive trait, such as this one, that you would predict there would be no color blindness in this person, because they have one good copy of this gene. Well, the X chromosome does something rather interesting. At some point in embryonic development, and we'll just draw here a 4-cell embryo. Each one of these has our 2 X chromosomes, as well as all the other chromosomes, but we're just going to look at the X chromosome. And they all are heterozygous. So all is good right now. But what happens here, early in embryonic development, is in a rather random manner, an RNA is produced by one of these X chromosomes. And this RNA comes from a gene that doesn't make a protein, it just makes this RNA. And it makes this RNA called XIST. What XIST does is whichever chromosome it's expressed from, it wraps around it. And it's random. And so let's draw this in a random manner then, where it looks like that. Now, every time these cells divide now, the X chromosome that is inactivated by this XIST gene will remain inactive. So when this divides into 8 cells, let's see if I can draw this here. this cell here will only have the positive gene expressed, the non-mutated one, and it'll maintain this inactive X chromosome. Likewise, this chromosome, as it divides, only the chromosome that had the mutation on it will be expressed. The other chromosome is still there, but it is represented as an inactive chromosome. And the same is true for the rest of these. So let's go ahead and piece them in. This one, when it divides, you're going to get your two mutated ones. And then we need to finish this one off here. And so now what we've done is we've artificially, so to speak, created some cells that only express the negative gene, the mutated gene, for color blindness. And so that means roughly half of the cells of this developing embryo, which will then become a fetus and then an adult, half of those cells will lack a functional gene for determining color in your vision. As this person eventually develops on to an adult, whether or not they are colorblind depends. It depends a lot on, as these cells differentiate, which ones ultimately come along here to make the eyes and the pigmentation in the eyes that helps determine color. If, for some luck, the negative ones are selected for that end up being the cells that determine eye color, then this person would be colorblind. However, if they got lucky and these cells went on to become the eye cells, then they would still be able to see color. So because of this random X inactivation, being heterozygous doesn't protect them necessarily. And because we're talking about epigenetics, the thing that is important to remember is that every time this cell divides, that same X chromosome is going to be inactive. So all depending on which cells these become will determine whether or not the trait of colorblindness persists. Now, this is true for any gene on the X chromosome, not just colorblindness genes, but any gene on the X chromosome. Lastly, I want to talk about this phenomenon called genomic imprinting. So as we begin genomic imprinting, let's consider these three genotypes. And we'll talk more about recessive and dominant traits later. But for now, you should know, or you may remember from a previous class, that individuals who are homozygous dominant, two dominant alleles, or one dominant allele, one recessive allele, these individuals will not have the mutant phenotype. However, this individual who is a, two mutations of the gene A, they will have mutant phenotype. And for decades, ever since Gregor Mendel, we assumed this was true, and moreover, we assumed that it didn't matter if mom or dad gave you which trait, meaning if mom gave you the big A allele and dad gave you the mutant little a allele, or vice versa, the result would be that you would not have the mutant phenotype. The source of the genes did not matter. But what genomic imprinting is now telling us is that it does matter which parent gave you the mutation. So to describe this phenomenon of genomic imprinting, a good gene to look at is a gene called IGF2. Okay, in talking about IGF2, let's look at this embryo here. And this embryo, this chromosome from dad, and this chromosome from mom. We know that each of these chromosomes contains this gene called IGF2. And in this case, let's assume that IGF2 is not mutated in either the chromosome that came from mom or the one that came from dad. So we'll put plus plus. However, what we know is that only the IGF2 that we get from dad is expressed. The IGF2 we get from mom is not expressed. Wild type or mutated, it doesn't matter. So in this embryo here, which is plus plus, IGF2 is expressed only from the dad's chromosome, but not from mom's. The gene IGF2 is important in regulating the size of the progeny and the size of the placenta. So this embryo here would be of average size, and the placenta would also be average. Now if you take another embryo, let's look at this embryo here, and we still have the chromosome from dad and the chromosome from mom. They both have the IGF2 gene, but the IGF2 gene from mom, on mom's chromosome that she gave us, is mutated. The one we get from dad is not mutated. However, this should not be a problem, because remember, it's only the dad's IGF2 gene that is ever expressed. Mom's copy is never expressed. So if it's mutated or not mutated, it doesn't matter. So this progeny, this child that would result from this, would be average size with an average placenta. I should mention real quick, the reason that it's not expressed from mom's chromosome, and you might have guessed this, is because of methylation. Its promoter, the DNA and its promoter, those cytosines, are methylated. So this one is also, of course, methylated. Now this embryo, let's look at this third example here. In this example, we also have a chromosome from dad and a chromosome from mom. They both have the IGF2 gene, but this time the one from dad is mutated and the one from mom is not mutated. However, this results in a low birth weight, small placenta, and in general, a sicker child. And the reason for that is because the only good copy of IGF2 you have, or this individual has, is on the mom's chromosome. And mom's source of IGF2 is not expressed. Dad's is expressed, but it's expressing a mutated version of it, so it's not going to help. Mom's copy would help if it could be expressed, but it can't be expressed. So these kinds of studies showed that it matters which parent gives us which genes. Not in all cases, but in these cases it is true. Okay, I want to erase this. I want to talk a little bit more about this genomic imprinting phenomenon. As I mentioned, that methylation is what causes it. But I want you to know that the process of making sperm, spermatogenesis, starting with a precursor cell and then through several events that we'll talk about when we talk about meiosis, will eventually give you a sperm cell. And during this process, certain genes are methylated. Just some genes. And likewise, during oogenesis, the process of making oocytes or eggs, again, you start off with a precursor. Through several steps, you end up making an oocyte. And just like with the production of sperm, different genes are methylated. Often, the genes that are methylated during spermatogenesis are not methylated during oogenesis, and vice versa. Not always, but often that is the case. And this is to result in a progeny which contains genomes from dad and genomes from mom that together result in an optimal size and health. Usually this does lead to an optimally sized individual. However, due to things like we saw with IGF2, sometimes it doesn't lead to an optimally sized individual. It can actually cause a problem. And I want to talk about a very interesting thing that happens on chromosome 15. Chromosome 15 receives different methylation patterns for different genes, with the goal of ending up with an optimally sized and healthy individual. But if this was chromosome 15, and in fact, let's draw two possibilities here. And remember, in these individuals here, some genes are methylated in different regions. If chromosome 15 is mutated from the dad, from the sperm source, we remove part of this chromosome, but just from the dad's copy, but not from mom's copy. And down here, we can talk about the reverse. When we mutate mom's copy, and so we're going to remove part of that chromosome because it's mutated. Now, the ones we removed now will result in different genes being expressed from mom or dad. And the interesting thing about this is, and let me erase what's over here so I can explain it to you. Okay, so as I was saying, the interesting that results from this is that two different diseases can occur if chromosome 15 receives mutations on the end of the chromosome from dad. it leads to a disease called Prader-Willi. Now, people with Prader-Willi, they'll suffer from an uncontrolled appetite that leads to obesity. They will have some behavior, cognitive problems, and problems in sexual development. Now, individuals who have the same set of genes removed on chromosome 15, but it's passed on from mom, will lead to a completely different syndrome called Angelman syndrome. People with Angelman syndrome don't suffer from these same characteristics. They tend to be hyperactive. Often they will have sleep disorders. And they have this almost contagious, happy disposition. They're always smiling, always seem to be happy. Same genes being removed, but depending on if those genes are removed from dad or from mom, results in two completely different syndromes with different characteristics. Okay, that ends this podcast. This was a pretty long podcast. We talked about a lot of different things. We talked about the definition of epigenetics, with the key being that it changes the gene expression without changing the DNA. We then talked about the molecular mechanisms of epigenetics, mainly thinking about DNA methylation and histone modifications. And then we spent the rest of the time talking about different ways that causes this change in mechanisms. We talked about behavior, environment, twins, X inactivation, and finally we talked about genomic imprinting. Okay, that ends this podcast on epigenetics and ends all of our podcasts on bacteria and eukaryotic gene regulation. If you have any questions at all, please make sure you come see me. If not, I will see you in class. Bye.
You're still here? It's over. Go home. Go.